in Mediterranean olive groves, and this research has been funded by EFSA. And we know that in Europe, spittlebugs are the most common xylem sap feeders, unlike in the Americas. And among these spittlebugs, we know very well that the most common, abundant, ubiquitous species is Philenus spumarius, which is also the major vector of Xylella in Europe. Very recently, also other species of spittlebugs like Philenus italosignus and Neophilenus campestris have been found to be competent vector, although they actual role in the field is probably of minor importance. And then among sharpshooters, the most common species we have in Italy is Cicadella viridis. And about the transmission, Nicola Bodino, a postdoc in my lab, will talk later this afternoon about the transmission competence of these species. The idea of the work was to provide a lot of information, data set on population dynamics, phenology, abundance of nymphs and adults, their host plant association, in order to be able to feed predictive model that can help in designing control measure that are aimed or at slowing disease progression in the infected areas or assessing risk in non-infected areas. For this, we choose 4 different experimental sites, olive grove agroecosystems that were situated in 2 distantly located regions of Italy, Apulia region, that we all know for Xylella, and Liguria, which is close to the Paca region of France in northern Italy. And in each one of the 2 sites, we selected 1 olive grove in the inland and 1 in the coastal area. I don't want to go too much into the detail of sampling methodology for the reason of time, but nymphs were, of course, spotted and counted by direct observation using kind of frame, transect. They were counted and with conservative sampling. We didn't took them. And each nymph was associated with its host plants, and sampling were based on weekly sampling. As for the adults, we use sweeping net, and also this sampling was conservative. And for the adults, we investigated 3 different vegetation compartments within the olive agroecosystem, the herbaceous cover, the olive canopies, and alternative woody hosts for the spittlebugs, shrubs and trees that are that were inside or surrounding the olive trees. Going to the results, here we can see the stage structured population. Each color corresponds to a different life stage. Stage structured population of Philenus spumarius nymphs, on the bottom in the Apulia region, on the top and on the bottom in the Liguria region. And although the 2 regions are quite distant, also different latitudes, we can see that basically the period of development really overlap, and nymphs hatch in early March and disappear by late May, globally considered. The same data represented in this graphical form help us to appreciate also the level of abundance of this nymphal population over the time. And so we can see that really nymphal population peaks in mid-April, more or less in mid-April, both in northern Italy and southern Italy. When we refer to, not to chronological date, but to phenological date, data. That means we try to calculate the degree days needed for the appearance of the different life stages. Data are not so constant, are different among sites, and the reason for that will be discussed in the conclusion. But this kind of data are not so constant as the chronological one. As for the adult population, here you can see represented in red the adults of Philenus spumarius that were collected on herbaceous cover, in green on olive, and in blue on alternative woody host. This is for the Apulia region, and we can see that adults can be found on olive canopy mainly from May until early July. Then adults disappear, virtually disappear from ground vegetation over the summer months. This is also because the vegetation becomes completely dry under these warm and dry Mediterranean conditions. And of course, shrubs and trees other than olives are really important alternative hosts, especially over the summer. As for the Liguria region, the picture is slightly different. And it's interesting to note that adults can be found on olive canopy basically all through the summer. And adults are abundant on ground vegetation through the wool period, spring-autumn, except for a short interruption during July-August, the most dry months. And also in these sites, shrubs and trees actually prove to be important alternative hosts for the vector. As for the other species, Neophilenus spumarius nymphs, this is the representation of the stage structural population of the nymphs of this species that was found both in Apulia and in Ligurian olive groves. And we can say that nymphal population follow a very similar trend to the ones of the nymphs of Philenus spumarius, although with a lower abundance. There is the development is a bit delay compared to the one of Philenus spumarius, but they really overlap. We found a decent number of another spittlebug species, Afrophoralni. This is the nymphal stage structural population, very different population level between the 2 years. These nymphs develop later compared to Philenus spumarius. We have population peak even on May. This species was found only in the Ligurian olive groves and never in Apulia. And the population level of this species was lower compared to Neophilenus campestris. So ranking for importance, by far Philenus spumarius has the highest population in olive groves, then Neophilenus, and finally Afrophoralni, if any. As for the adults of Neophilenus, we have this pattern in population dynamics in the 3 vegetational compartment. And we can see that these adults were mainly collected on the herbaceous cover, still associated like nymphs to grasses, Poaceae. The adults of this species were virtually absent in the olive agroecosystem in all the vegetation compartment in the summer months. And here we are talking about the Apulia situation. The highest abundance of Neophilenus campestris adults were registered early in the season, in late May. As for the Liguria situation, the population abundance is a bit has a different distribution. Also here, the adults were really associated with gramineous plants. Also in Liguria, we failed to collect adults of this species during summer months in the different vegetation compartment of the olive agroecosystem, but the highest abundance of adults of Neophylenus were registered late in the season during autumn. As for the Afrophora alni that we only found in the Liguria region, most of the adults were collected on woody hosts, including olive canopies, but also oaks that this species like also Philenus like oaks a lot. And the highest density, although these densities were not very high, but were registered in June, and then they decreased during the summer. I would like to point out an interesting feature about the abundance of spittlebug adults on olive trees over the season, which is differential between Apulia and Liguria region in relation to the phenological stage of olive trees. In Apulia, we observe a good number of adults we collected on the canopies of olive only for a restricted window of time from May, as soon as they emerge as adults, until the early July, in correspondence of flowering and the very beginning of fruit development of olive trees. While in the Liguria region, the presence of adults of Philenus on olive canopies is more extended, and they are present well beyond the flowering period and the whole fruit development, and even during fruit maturation. The potential epidemiological consequences of this presence on the olive is quite clear, I guess. As for the host plant association, I underlined in green the Fabaceae and in red the Asteraceae plants. And you can see that in the Apulia olive groves, most of the spittlebugs nymphs were associated with Fabaceae, while most of the nymphs in the Liguria region were associated with Asteraceae, plants in the family Asteraceae. This host plant selection is also driven by the different floral composition of the ground cover of the olive groves, in the sense that there were more Fabaceae in the olive groves of Apulia and more Asteraceae in Liguria. As for the host plant association of Afrophora alni nymphs, we found that they prefer, or at least they select, mostly Asteraceae. And as for Neophylenus campestris, both in northern and southern Italy, this species is really strongly associated with plants in the family Poaceae, with gramineous plants. I just want to remember that results that are consistent with the one that we obtained in this research have been found in another field-based research conducted in Apulia region in a high number of olive groves that were located both in the infected areas and outside the infected area. And then I go to the conclusion. We can simply conclude that the phenological pattern in the two regions is similar if referred to chronological time, but is different if we refer to physiological time. And this can be due to a non-linear component in the temperature development rate function of Philenus spumarius. The population density was higher, that we observed was higher in the olive groves of the Liguria region compared to the one in Apulia, but these one were located in the Bari area, not in the Salento infected area. And then we confirmed the high polyphagy of Philenus spumarius, and we noticed that Afrophora alni has a similar host range of like the one of Philenus spumarius, while Neophilenus is strictly associated with Poaceae. And then we observed this extended period of presence of adults of Philenus in olive canopy in Liguria compared to the Apulia. And then with this, I conclude, because time is over, and I want to thank EFSA for funding this research. All the data are included in the final report, which is published on the EFSA journal, and I want to thank you for your attention.